to say good afternoon to everyone. We say a very big welcome to you to the second edition of the World Phage Week, IBRT, Ibadan WPW Research Team World Phage Week. And thank you so much for joining us. At the moment, on our last count, we had about 180 registered participants, and hopefully we get to have more because we plan to share the YouTube link and the Zoom link, well, to as many individuals who, perhaps could not register. And then, of course, because of the caliber of the person, the persons that we are having for this year's webinar, this is one program that every Phage enthusiast all around the world will want to be a part of. So I am a minute behind my time schedule. But before I allow my mentor, my dad, the faculty head of Ibadan WPW Research Team, to go ahead with the proper introduction, the welcoming of all of the registered participants, and of course, special greetings from the Premier University of Ibadan to Professor Graham Hatfull. I would just want to say a very big thank you to all of our sponsors, to all of our partners. I think, Jessica, you can help me to move the slide. So you're welcome to this year's Assessing the Readiness of the Global Phage Community in Combating Antimicrobial Resistance. And we want to say a very big thank you to all of our sponsors, Phage Directory, for the full-fledged technical support, with the Zoom link, with the YouTube link, with the emails, with the website. I can keep mentioning as much as I could, but because of the paucity of time, I would just stay on that. So we want to appreciate Phage Directory. We also would want to Then we also would like to appreciate Baylor Taylor Laboratory. I am not sure if Dr. Sabrina Green is on the meeting with us at the moment, but big appreciation to Baylor Taylor, for the financial support, and then also for keeping track, with the preparation towards this meeting. Okay, Sabrina is here. So thank you very much, Sabrina. It's really, really nice to have you. We also would like to say a very big thank you to Phage GENOMICS company. I don't know if Ben Ouch is here with us on this meeting. Big thank you to Phage GENOMICS. We also would like to say a very big thank you to the Indian Bacteriophage Research Consortium, headed by Professor Umi Bajpai. Thank you very much. And then also we want to appreciate African Phage Forum, led by Dr. Emmanuel Nadi. We say a very big thank you to each and every individual that has put forward, their support to ensuring that this year's program is a huge success. Also, we would want to appreciate SEAPHAGES. We want to appreciate HHMI. I remember we sent a mail of notification, to them that one of the SEAPHAGES team in University of Ibadan is having a program, and we are inviting, our very own Professor Graham Hatfull. And immediately we did that, it was a very welcoming gesture from, the handlers of the HHMI SEAPHAGES email. So at this point, I would want to keep quiet and then, allow Professor Oji Ademowo, the founder of Ibadan Bacteriophage Research Team and the faculty head of Ibadan Bacteriophage Research Team, to welcome each and every person to this year's World Phage Week, and then have a very warm welcome to Professor Graham Hatfull himself. Thank you very much.
Yeah. Good day, everybody. I'm sure you're hearing me. So I want to. Are you hearing me?
Yes.
Yeah, thank you. Okay. I want to welcome everybody to this all-important meeting of today. I am particularly delighted about the huge support received from everywhere, both the partners and the sponsors and the speakers. We look forward to a beautiful exercise today, and so that I do not waste most of our time, I really want to, on behalf of the IBRT, the Ibadan BACTERIOPHAGE research team, I want to welcome you all to this meeting. And I also want to welcome all participants from around the world who are listening to me now. We cherish all of you, and we wish all of you well, and we hope for useful deliberation. Thank you very much. Thank you. We appreciate you. You're welcome.
Thank you very, very much, sir. That was the voice of Professor Oji Ademowo, the faculty head of Ibadan Bacteriophage Research Team. And well, hopefully we would have other members of the faculty team working with HHMI SEAPHAGES to ensure that the SEAPHAGES program here in University of Ibadan is growing stronger and better. Thank you very much, sir. We are still expecting other members of the faculty team to join us. So without further ado, I find it necessary, for us to delve right into the program of today. So for session one, we have, well, will I say, well, quite appropriately, we have one of the biggest and one of the best bacteriophage researchers, one world-renowned researcher of great repute, Professor Graham Hatfull, joining us. Now, of course, everyone seems to know Professor Graham Hatfull's profile, but I think it's just necessary that I read it out to every participant again so that we will delve right into the talk. So Professor Graham Hatfull is a professor of biological sciences at the University of Pittsburgh. He received his PhD in molecular biology from Edinburgh University in the year 1981 and did his postdoctoral work at Yale University with Dr. Nigel Grandley and at the Medical Research Council at Cambridge University with Fred Sanger and Bart Perel. He joined the University of Pittsburgh in 1988 and served as the chair of the Department of Biological Sciences from 2003 to 2011. Dr. Hatfull's research focuses on the molecular genetics of the mycobacteria and their bacteriophages and their use for educational and clinical uses. Dr. Hatfull has published over 250 peer-reviewed articles, 43 chapters or reviews, and has co-edited 4 books. He's a fellow of the American Academy of Microbiology, a fellow of the American Association of the Advancement of Sciences, and a member of the American Academy of Arts and Sciences. He has won the award. He has been an Howard EU Medical Institute professor since the year 2002. Distinguished ladies and gentlemen, registrant participants from all over the world, I say a very warm welcome to Professor Graham Hatfull, and thank you very much, sir, for joining us. So the floor is yours, sir.
Thank you very much. Let me just navigate sharing my screen here first. I'm assuming you can hear me okay. And so first of all, let me say a big thank you for the invitation, for your very kind introduction. It's an absolute delight to be here and to share some of our stories and interests in phage research and phage applications. I have quite a few topics I'd like to walk our way through. So I'm going to do that all relatively quickly. Hopefully we'll have time for a question or two at the end. But please don't hesitate to contact me afterwards by email with questions or go dive into some of the literature for more details on topics that you might be interested in. So our interest for a long time now has been on mycobacterial diseases, and there's really 2 main categories, I would say. The first is tuberculosis, disease caused by Mycobacterium tuberculosis. It infects a large part of the world's population, 10 million new cases of TB a year, many drug-resistant strains, and lots of challenges in how to control TB at a global level, including a response to the extensive antibiotic resistance which has been observed. The second is the sort of the general classification of the NTMs, the non-tuberculous mycobacteria, which cause also serious infections, but are more constrained in terms of their distribution. So Mycobacterium abscessus infections are quite common in cystic fibrosis patients. These are predominantly pulmonary infections like TB. They're a strong contraindication for lung transplantation, and for patients that have had lung transplants, M. abscessus infections can be a serious problem because of disseminated disease, in part in response to the need for post-transplant immunosuppressive drugs. These strains are often highly resistant and refractory to treatment and cause very serious health problems. So 2 different types of diseases, and I'm going to talk about the phages of the mycobacteria, but I want to kind of cut to the chase with a brief summary. So this will provide a context of the rest of much of the rest of the talk. And the challenges and the opportunities are clearly distinct for TB and for NTM infections. In the words of Dickens, we have the best of times and the worst of times. For TB, the good news is that there's limited genetic variability of TB. There's rather little variation in phage infection profiles, which means you have the prospect of coming up with a small phage cocktail that could be used for essentially all MTB infections without having any need for personalization. The problem, however, is that there are real questions as to how well phage therapy will work, given the intracellular environments of the bacteria, bacteria being walled off in granulomas, etc. And so access becomes a fairly major question. For the NTMs, the good news is that there are a number of desperate compassionate use cases, and the interventions with phages that have happened to date show a strong safety profile in humans, and at least in some cases, really good but anecdotal efficacy. And unlike many other bacterial infections, phage resistance does not appear to be a major concern, at least as far as we can tell at this time, either in vitro or in vivo. However, the downside is that these NTM strains vary enormously in their genetics, and they vary enormously in their susceptibility to the phage infection profiles. So what phages might work for one patient don't work necessarily for another patient. Personalization is required. There are not phages for all strains that have been isolated, and those questions clearly need to be addressed prior to any broader utility. So I hope that'll set the stage, and I'll tell you a little bit more about phages and then our experiences with using them therapeutically and what we think the prospects are. So the phage ecologists have told us that this is an amazing population of phages in the natural world, 10 to the 31 phage particles estimated in the biosphere, with 10 to 23 phage infections per second, and a very, very old population. And it's because of these parameters that we are forced to understand that this is a highly diverse and highly genetically variable population of viruses. And so understanding that question, the diversity of the phage population, is a question we've been interested in for some time. You can imagine addressing that question using metagenomic approaches, where you just collect phages from the environment, concentrate them, sequence the DNA, compare the sequences, and fill up your hard drive with this massive data sets. An approach that's been very informative, especially about dynamics and broader questions of diversity. The other approach, however, is to do it as a genome by genome, phage by phage approach. The disadvantage is you don't get quite as much raw data, so you don't fill up your data, your hard drive so quickly with all of that data. But the good news is that you also have the individual phage isolates. You have stuff in the computer and you have stuff in the freezer. You have all the isolates in the freezer, each of which can be propagated, grown, manipulated, studied, engineered, and of course, potentially used therapeutically. And so that is the approach that we've taken, and I'll tell you a little bit more about. So isolating bacteriophages from the environment is relatively simple. It's been done for 100 years. You need to have a particular bacterial strain that you can grow and form a bacterial lawn on solid media, a sample from anywhere in the environment that you or your students may choose to go and sample, and to make a simple extract and plate it out and look for plaques, each of these being an area of infection caused by an original single phage particle that has then infected a cell, replicated, and reproduced itself. And so this plaque assay is enormously useful and has been widely used and is robust and utile. But you have to pick a host in order to do this process. So it very much depends on which bacterium you use. And we started many, many years ago using a bacterium called Mycobacterium smegmatis. Why? Because it is a close relative of these pathogens, Mycobacterium tuberculosis and Mycobacterium abscessus that I've already mentioned, but it itself is a non-pathogen. It grows relatively quickly and much faster than Mycobacterium tuberculosis itself. So it's at least reasonably easy to handle in the lab, and that can be used to isolate phages. And at least when we started out, there was the assumption that some of these phages may also infect other bacteria, including these pathogens. And even if they didn't, what we learned about the phage biology from this smegmatis phages would be useful as tools for developing genetic systems, perhaps diagnostic systems, perhaps strategies for vaccines for these pathogenic strains. I'd just say parenthetically that we've actually been involved in the isolation of phages using a number of other hosts, but all focused within the Actinobacteria phylum. So the smegmatis phages are the ones I'm going to focus on today. So if we want to understand genetic diversity of the phage population, we don't want to have one or two sequence phages. We want to have large collections of phages so we get a broader view. And who's going to do all of that? We've made progress by the development of these integrated research education programs where we can use phage discovery in genomics to introduce students, young students, high school students, first-year undergraduate students in discovering phages using these approaches and characterizing their genomes. And so there are several of these programs that we've been involved in. By far the largest, as was mentioned very, very nicely in the introduction there, was the SEAPHAGES program. These are all acronyms, so it has nothing to do with the oceans. C is an acronym for the Science Education Alliance, which is an office or a component, if you like, a department within the Howard Hughes Medical Institute. And then the phages is the Phage Hunters Advancing Genomics and Evolutionary Science. So this is a program that started for introducing the young students to research experiences, started in 2008 with 12 participating institutions. More have been added each year. They are primarily in the US, but have representations, of course, from Nigeria and as well as Canada, New Zealand, and Mexico. The types of institutions vary enormously, and this is a key part of the structure because you don't need to be an expert or experienced in phage biology or in phage isolation in order to be able to go through a process of training and then to teach students how to do this and to advance our understanding of phage biology. And this is actually, we think of it as an example of a more generalizable programmatic structure that we call an inclusive research education community, or an IREC. And just in brief, the point is that there is centralized program administration and scientific leadership. There's a particular set of programmatic functions that are all support the overall endeavor. And then it is a process in which individual institutions can participate, can take advantage of these centralized parts to implement the course with their students at their institute in a way which works with them with support and guidance and with community sharing of information. In the US, this is often, I would say typically offered as a 2-term course for first-year undergraduates, with the first term being the most of the microbiology, usually offered in the fall or in the autumn, with a student commitment about 4 hours per week. So it's not a huge commitment, but it lasts over 2 terms. So the first term is the microbiology, where the students are isolating the phage, purifying them, amplifying them, looking at them in the electron microscope. isolating DNA for DNA analysis. And then the genomes are sequenced between these two terms, actually right around now in the academic year, at least in the US. And then the spring term is mostly the bioinformatics and involves genome annotation, gene identification, genome comparison, and understanding sort of evolutionary relationships and contexts, and then hopefully publishing this new information and these new insights. There's a lot of flexibility in the specific structure, depending on the institution, obviously whereabouts in the world, the time frame, the kind of semester structure that you're on. But there's a lot of flexibility in the way this works, so it can accommodate those types of alternative configurations. Because the information that's gleaned is all disseminated, we have a database and a website that seeks to consolidate that information and to collect it from the participating students and institutions, and then also make it broadly available to everybody. This is just a snapshot of the homepage of the website. It's at Phages DB. If anybody is interested, it is publicly accessible. There's some stats here on the homepage of, let's see if I can get them up here. The total number of phages that have now been isolated is just a little bit shy of 20,000. I think that's an impressive collection. We haven't sequenced all of those genomes, but over 3,800 have been sequenced, moved into GenBank, and we're going to focus today here really just on that subset which infect the Mycobacterium. So about 10,000 of these phages have been isolated on the Mycobacterium, principally Mycobacterium smegmatis, and about 2,000 or a little over 2,000 of those have been completely genomically sequenced and annotated. They've clearly come and have originated from around the world, although with predominance because of the development of the program primarily in the US, but I think with quite impressive coverage across the US for these different types of phages. And you can see, as we look at Africa, some nice phages here from our colleagues and friends in Nigeria, from South Africa, where we've run similar types of workshops, and some from Madagascar. We would like to populate this with phages. We'd like to populate the map with phages to similar density in Africa as we have in the US. I think we would learn an enormous amount about the phage population if we were able to do so. Just a quick look at morphology. Essentially, all of the phages isolated on the Mycobacteria are double-stranded DNA tail phages, either those with contractile tails, which is actually a minority of the phages. By far the majority are of the so-called cyphoviruses. They have these long, flexible, non-contractile tails. This one is particularly long and impressive. Most of the phages have isometric or soccer ball type shapes of heads. A few have these so-called prolate or elongated heads. And this is one of our favorites that one of our students in Pittsburgh named this phage corn dog because of its clear resemblance to the American delicacy as shown here. What do we learn about genomics and diversity? Well, first of all, they are indeed enormously diverse. And so I'm just illustrating part of the conclusion here from the diversity, which is that we can take these phages, and some of them are related to each other and some of them are not. Those that are related to each other, we can put into groups. I won't go into the specifics, but we call those clusters. Some of those can be subdivided into subclusters. And in this representation, I've just taken one example of each of the clusters and subclusters from all of the actinobacteria and compared them by comparing the genes that they shared gene content. And you can see how these all emerge from the central point here, illustrating the fact that most phages, when you compare in this way, end up actually sharing, most pairwise comparison of these distinct groups end up sharing little or no genetic information at all. How does this happen? If we start looking in more detail at some of these genomes, I'm showing you some just a few examples from a few of the clusters. The genome maps are aligned. The genes are shown here. They're rather small boxes. Clearly you can't read them, but you don't need to. Between the genomes, there's a pairwise representation shown in this coloring here of DNA sequence similarity. And what you can see is that the characteristic relationships are regions, it's shown in the purple, that are very closely related, interspersed by regions of either a more reddish color or no color, where they are different DNA sequences. And so as you do these comparisons, you see this abundance of regions of interspersed similarity and lack of similarity. And it appears as though these have been constructed essentially mosaically, with different pieces being exchanged between segments, and that reflecting how you can generate the overall diversity of the phage population. In essence, phages are constructed the same way that the ancient Romans made their mosaic art, where they took stone tiles, in this case, from different geological origin, different colors, and then could take these stones and could assemble them together into whatever picture that they wanted. And you could indeed remove all of these stones and mix them up and use them to make different pictures by using them in different combinations and different configurations. And phage genomes are assembled in essentially exactly the same way. And so this is just a further but more detailed example of that, where I'm showing a small segment of several different phages that are unrelated at the DNA sequence level, but then looking to see what genes they share. So here's a phage called Fran, here's a phage called corn dog that I mentioned previously. Most of these genes are different because you can tell by the different colors for the most part. However, this gene and this gene are related at the amino acid sequence level and have a common origin, and yet each of these genes is in a distinct genomic context. The sequences to the left and to the right are different in each case. And so this gene is like a mosaic tile, which has found itself in different genomic contexts in these genomes through a mechanism that we presume predominantly involves illegitimate DNA recombination, putting sequences in new contexts. And then there are many, many more examples of that, as you can see here, these links between these genes. So this genome mosaicism is the common theme that we see when we compare phage genomes, and it can account for the incredible diversity that we see simply because of the ability for, over a long period of time, in a dynamic set of relationships, for genes to be put together in novel combinations by this mechanism. I want to turn now to how we can take this information and how it's been useful, and in fact, I would argue essential, to go about the process of thinking about the use of phages therapeutically. Our interest in this started when we got a message from a colleague of ours in London in late 2017, essentially telling us that they had 2 patients with rather similar profiles. They were young female cystic fibrosis patients. They had both had double lung transplants, were taking immunosuppressive drugs in order to support the new lungs that they'd received, had done very well with it, except had then gotten these Mycobacterium abscessus infections, which had disseminated and caused great health risks. And these strains were highly antibiotic resistant. And even though they've been treated with large repertoires of antibiotics to try to control them, they were essentially unsuccessful, and they were down to their last resort. They ran out of options. And the question that our colleague asked me was, are there phages that could plausibly be used as a last resort? And so we set about trying to look for phages for treating these kinds of infections. This just shows what one of these infections looks like for one of the patients. The scan shows these quite extensive infections. There's a nasty infected node here in the liver, and you can see these skin nodules from which Mycobacterium abscessus could be cultured, and then the sternal wound from the transplant failed to close and was infected with Mycobacterium abscessus. And this Mycobacterium abscessus could be cultured from these and grown in the lab, and that's the strain that was sent to us. And you can see this long list of antibiotics as well as immunosuppressive drugs that these patients were taking. Now, the immediate problem that we had was that very few, if any, phages had been isolated specifically on Mycobacterium abscessus. And so there was not a lot of M. abscessus specific phages to go for. And as I'll show you, there's actually this huge variation in strains. So even if there were phages isolated for one strain of M. abscessus, it doesn't mean that they would infect other strains anyway. So that was a very limited set of access that we had. And it remains true that very few phages have been isolated directly on Mycobacterium abscessus. But we were in a position where we could turn to our large collection of Mycobacterium smegmatis phages, of which we had 2,000 that have been genomically characterized. And because we knew about the genomic relationships and we had some insights into host range, we could actually whittle this down to a rather smaller number, 100 and perhaps 200, that we could screen to see if any of these had host ranges that extended from Mycobacterium smegmatis to Mycobacterium abscessus, strain GD01, the strain from one of these particular patients. And in fact, we were successful in identifying 3 phage candidates. But before we could use these therapeutically, we had some more work to do. And so in order to explain that, I need to take a short detour through thinking about some of these phage life cycles and to understand the challenges that were presented. So first of all, most mycobacteriophages are what is called temperate, more than 50%. It is very common to find temperate phages. Temperate phages differ from lytic phages in that when temperate phages infect their host, the DNA is injected and a decision is made. Maybe 80% or something of the time, they go through this normal process of lytic replication and cell death, phage production, and the cell is dead. But about 20%, it can be a higher proportion of the time, the genes that are required to do this are switched off. The cell does not die. The DNA circularizes and then usually integrates into the bacterial chromosome, where it is then called a prophage. And that prophage can sit there happily for many, many, many generations. And this cell survives the phage infection and in fact survives reinfection by the same phage in a process known as superinfection immunity. So lytic phages are highly favored for therapy purposes because they kill the bacteria efficiently. Temperate phages have a high level of survival and therefore are not useful unless you do something about it. And in fact, apart from one of the phages, most of those that infect the M. abscessus strains that we tested are in fact temperate, and so we needed to do something about that, and I'll show you what we did. However, before I leave this topic of these phage life cycles, there's another important aspect that I want to introduce, and I think you'll see why in a few minutes when we come back to talk more about what we've learned about the Mycobacterium abscessus strains. And that is that these prophages shown here that exist naturally, many bacterial strains carry one or more prophages. These are not just sitting there neutrally doing nothing. It is very common for them to have genes that are expressed in the lysogen that specifically confer viral defense, meaning they're used by these strains to fight off other types of phages, other types of viruses that are otherwise going to be trying to attack them in the environment. And I'm going to illustrate that with an example from a paper we published, Gentile et al. A couple of years ago, just illustrating this point. And so let me explain this slide. Here is showing the representation of a lysogen called SPASH. Here's another one called G9C. So don't worry about this. I'm going to focus on SPASH. So this is a phage that we've used. It's a temperate phage we've used to form a lysogen. And then what we've done is to measure the efficiency of plaquing of individual phages on a SPASH lysogen relative to a non-lysogen. So we just take the two strains. One has a prophage, one doesn't. And we can compare how well these phages infect. And we can do this with many, many phages because we have such a large collection of phages, all that infect the same common bacterial strain. So most of these phages don't care about the prophage. The efficiency of plaquing is 1. They give an equal number of plaques on this SPASH lysogenic strain as to a non-lysogen. However, as we look down our list, we see, well, first of all, we see this exception here. Now, this is the inability of SPASH to infect itself. That's the process known as superinfection immunity. However, this lysogen also defends to many orders of magnitude against infection of, in this case, Crossroads, or in this case, Phage Giles. And in fact, we know that SPASH encodes two separate defense systems that are influencing how the phages infect these strains. So this is just an illustration of a general property of how resident prophages in bacterial genomes dictate the phage infection profiles in a much more broader, impactful way. Now, so just bearing with that in mind, we now are going to return to how we're going to engineer these genomes in order to make them lytic as opposed to temperate. We know what genes are involved. We simply need to go and engineer them in order to knock out the relevant genes, usually the repressor and the integrase genes. We developed a couple of approaches for doing this. Some time ago, we described a recombinering-based method for engineering these phage genomes. And more recently, we've added a second component, which is a CRISPR selection or a counter selection, which helps to increase the efficiency with which the mutants can be constructed. And so you can read about these more in the Wetzel et al. Paper that was recently published, but these methods generally make it relatively simple to manipulate phage genomes so that we can make them therapeutically useful. So we ended up being successful in isolating 3 phages. We thought we would need to use 3 phages in order as a combat against resistance. One of these phages was Muddy, a phage isolated in South Africa that didn't require any further action. Zoe J, which was isolated at a SEAPHAGES school in Providence College, and which was engineered in the way I just described by Kathleen Cornely when she visited on sabbatical at the University of Pittsburgh. And then lastly, BPS, which was also engineered to be lytic. And we used genetics to isolate a host range variant. Think of this like a COVID variant that now efficiently infects the Mycobacterium abscessus strain. So at least for this one patient, we were all set to contemplate a therapeutic intervention. Unfortunately for this other strain, we didn't have successful isolating phages for the most part, and this patient actually died a couple of months or so after we started looking, which at least I think helps to emphasize the severity and the lethality of these kinds of infections. So we grew these phages to high titer and purified them to exquisite homogeneity using these equilibrium density cesium chloride methods, old but very effective methods. And then we went about to test whether these phages do indeed efficiently kill the bacteria in the lab. And we did this by just setting up a range of combinations of different numbers of bacterial cells and different amounts of phage. We incubate those in liquid culture and then spot them onto solid media for survival. And so this is cases where you can see the bacteria are growing here because we didn't treat them, and these ones are all killed. All 3 phages work quite well, some somewhat better than others, but the mixture together is quite impressive at killing over a wide range of cell and phage concentrations. So these phages were administered intravenously, as well as some topical administration for the skin nodules and on the sternal wound. They were administered about 10 to the 9 PFU per dose twice daily. And the good news is we saw no adverse reactions, extremely well tolerated. And in post-treatment bacterial isolates, we have not seen resistance to the phages either, which we're also encouraged with. Within 6 weeks or so, there was substantial clinical improvement, and it's just illustrated here by these scans, by the resolution of this infected node in the liver, the resolution of the skin nodules, and the healing of the sternal wound. So we were very pleased. It's a very positive clinical outcome with making a very substantial clinical difference. Will this work for other patients with similar types of M. abscessus infections? The question remains unanswered at this point. The key is the matter of specificity. And we see specificity as this double-edged sword. On the one hand, the ability to use these precision missiles to go and take out bad bacteria while leaving all the good ones perfectly intact is extremely attractive. On the other hand, the specificity can be so great that we have this problem that phages that infect one strain won't necessarily infect other strains. And we've received something like 200 clinical isolates of these strains from physicians who are out of options for patients with these types of infections. It's clearly a serious problem on a global scale. And so we have sought to try to find phages. These M. abscissa strains come in different colony morphotypes. There's the so-called rough and the so-called smooth. We have no good therapeutic phages for these smooth strains, but we think we have at least one for about 75% of the rough strains. So we're beginning to narrow this down to see where we can have some therapeutic utility. And I'm not going to go into all of the gory details here, but I just want to illustrate that these are some of the strains. There's only about 80 or so here. Their phylogenetic relationships from the conserved sequences here are displayed. Here's a summary of these phage infection profiles. And I just want to point out that these don't correlate in any obvious way to the genomic relationship. And so it is the phage infection profile is highly varied, and it's completely unpredictable from what we know about the genomic sequences of these strains. And if we just focus in on these strains that are very closely related to each other here, they all represent a single major clade, and yet these phage infection profiles are clearly highly varied. Now, so you may ask, well, what does determine the phage infection profiles? And this is a cause for coming back and thinking about what's going on in these bacteria and in their genomes. And in fact, what we now know from the genomics is that they are riddled with naturally occurring integrated prophages. And this is just illustrating some of the distribution. And you can see these prophages also have these unpredictable patterns. We don't have direct evidence yet that these prophages are specifically influencing the infection of these Mycobacterium smegmatis phages, but I think it's pretty certain that they do play a role, although the plasmids may play a role as well. So there's lots to be dissected in these patterns, and that will be the key to broadening this for other patients. So I want to just finish by thinking about what the prospects are for using phages to control Mycobacterium tuberculosis infection. In many ways, we're very encouraged by the compassionate use interventions for Mycobacterium abscessus. I showed you the one in detail. There are several others that we've been involved in, a number of which we see good outcomes. Can those good outcomes be extended to Mycobacterium tuberculosis? We know we have phages that infect TB, and I'll explain that in a little bit more detail. There's much less strain variation amongst TB clinical isolates, much less so than I just showed you for Mycobacterium abscessus. However, compassionate use cases, which whilst anecdotal in nature, can be very informative about certainly about safety issues and about clinical responses. They're very hard to come by for TB patients for a whole set of reasons that I won't go into, but it's very rare to find justifiable cases where you can get regulatory approval for compassionate use for TB. Clearly, we think there's a need for clinical trials, and we hope that that we can help to pave the way for this to happen sooner rather than later. We have tested now over a number of years different types of phages that have emerged, smegmatis phages, and tested them for the ability to infect TB, primarily using this lab strain that's very commonly used called H37RV. And all I'm showing you here is really an update on that, where we've taken any of the phages that have been isolated more recently and tested them on this in order to have a fairly good confidence of which phages can infect TB and which ones don't. However, that's just H37RV, the lab strain. The question is, what about clinical isolates? So we obtained from Sebastian Gagnon the reference set of TB strains. They're indicated on this phylogeny here. The phylogenetics of TB has revealed these major lineages. They're relatively closely related in terms of just branch lengths, but we have examples of each of these lineages that we can now test the phages on and ask, do the phages that infect the lab strain also infect the clinical isolates? You remember, this is the problem with Mycobacterium abscessus. Is it a problem with TB? Well, what we find first is that we have some phages. Here's one called Fionbath. It's been engineered to remove the integrase and the repressor genes I just showed, and it infects H37RV, that's shown here, but it also infects all of these other strains that we've tested. So there are phages that infect all the strains that we've tested, and we think that's extremely encouraging. There's some caveats though. Here's one phage. This is the BPS phage. I described this one to you previously, and I described that we had a host range mutant that we used that infects the Mycobacterium abscessus strain, and we used it therapeutically. Here we have a host range mutant to BPS that infects H37 very well, and it's very nice. However, that mutant, nor the parent, efficiently infects any other TB strain that we've tested. So that alone is not useful for us. Maybe there are other host range mutants of BPS that could be generated, but that's was a bit of a warning lesson for us in examining this. You cannot assume that a phage that infects one strain of TB necessarily infects other strains. And then I just want to point out this last one here. This is our good old friend Muddy that I also mentioned previously. Nicely infects H37RV. It infects some other strains, but not all strains. So that's an intermediate kind of phenotype. What can we do about it? Well, we can use our same strategy as we used previously. Go in and see if we can isolate host range mutants that have acquired the ability to infect one or more other TB strains. We've now done this with phage Muddy. We've isolated 3 new host range mutants using 2 different particular host strains to isolate them on. Each of these had single point mutations in a tail fiber gene, gene 24 in Muddy, as shown here. And in fact, all of these mutants now efficiently infect every TB strain that we've tested. And so we've used these genetic approaches as well as the engineering to expand this repertoire of useful phages for TB. And this brings us to a point where we can map this profile of different strains in different lineages of TB and how well they infect and kill. And so here's just a boring table that summarizes all of that. But it's the key to us putting together a small cocktail. The one we tested here was a 5-phage cocktail. It may be possible to use ones with as few as 3 phages. And if we just focus on the lower panel here, where it's the same kind of assay I showed you previously, different strains of TB mixed either without phage or with phage, in this case, the 5-phage cocktail at different phage concentrations, and then plated out for survivors. We see really very good killing over a broad range of cell and phage concentrations for these TB strains, widely across these clinical isolates. And we think that this or a similar cocktail can be used without personalization for intervention in TB infections. We hope to be able to try this in compassionate use cases where they might be available, and to propose this for future clinical evaluation. So that's where I'm going to conclude today. I'll just briefly summarize by saying that the SEAPHAGES program, I think, has been extremely powerful in identifying and discovering these diverse mycobacteriophages. I'm still impressed by the incredible diversity amongst these 2,000 phages, all of which were isolated against a single isolate of Mycobacterium smegmatis, and just very much favor extending this to this approach to other strains and other pathogens. I think these phages and their genomic diversity and their host range have been incredibly helpful, I would say essential, in moving forward with I think good progress towards therapeutic applications in these compassionate use cases. And clearly we have good outcomes for many Mycobacterium abscessus infections with a great safety record. I didn't talk about it, but a potential concern, especially if wanting to give extended IV administration, is that patients don't always, but can get a strong antibody-mediated immunity to the phages, which then neutralizes their activity. I didn't talk about that, but we have experienced one case where clearly this appeared to be an impediment to treatment. But there are other cases where patients have responded similarly, where the clinical outcome appeared to be quite good. And so we have a lot to understand about how the body reacts to these administrations and how we might be able to accommodate it from a therapeutic point of view. I think the prospects are quite exciting for the therapy for TB. I think this is in the category of potentially high risk, high return, high reward. The risk is that because of the issues that I mentioned at the very beginning, in terms of getting access of the phages to the bacteria in a clinical situation, may be an impediment that means that TB therapy, that the phage therapy for TB will never really be that effective. On the other hand, I think we probably won't understand more about that until it is evaluated either in compassionate use cases, in clinical trials, or in appropriate animal models if they are available and can be used in this way. There's much that we don't know about these therapeutic interventions and that we can't learn from compassionate use cases. What's the best route of administration? What's the best dosage? What's the regimen? What's the interaction with antibiotics? Are there interactions between the phages? There's much that we don't know, but I think that there's much to be learned in the coming years, I think, for these phages for mycobacterial infections and indeed for other pathogenic bacterial infections in general. Let me say a very big thank you to all of you for your attention. There's more people to thank that I can imagine. The people in yellow here are those who have played more direct roles, especially in the therapy aspects of what I talked about. All of the SEAPHAGES faculty and all the SEAPHAGES students have been absolutely tremendous and are an essential part of this story. And of course, our phage hunters everywhere. Their efforts, each individual one, represents a part of this story. And so I say a very big thank you to all of you. This reminds us that at times we do get together in person to celebrate our science and our study of phages. This was indeed a couple of years ago, but we hope that we will be back having in-person meetings in sometime in the not too distant future. And I think there are a few minutes, and I'd certainly be more than happy to answer any questions that you have. Thank you.
Okay, thank you very, very much, sir. Thank you very much for a very enlightening discussion. So in the course of your talk, some questions came in, and I really hope we'll be able to take all of these questions. Okay, so I will start with a question from is Dios. He said, thank you very much, Professor Hatfull, for a great talk. Regarding phages for Mycobacterium tuberculosis, I have worked with some phages using clinical isolates of Mycobacterium tuberculosis, and from my experiments, it appeared isolates obtained from sputum were more susceptible than those obtained from samples outside the pulmonary compartment. Could this be a one-off result, or there could actually be a pattern worth exploring? I would like to know your expert opinion on that. So can you help us with that answer very quickly so that we'll move over?
Yeah, no, so it's a really good question. And I think that you know, for the most part, we have in practice, taken isolates of bacteria and have tested them for phage infection lab, and then have extended that into a compassionate use intervention. However, we know very little about what the in vivo phage sensitivity is to the in vitro phage sensitivity, and bacteria that are in a clinical setting may be either more or less susceptible to phage infection than a strain that's grown in the lab. It's true for TB, I think. It's true for every pathogen. It's hard to know, unless you have really good animal models, it's hard to know what's going on in the in vivo situation, and you really don't learn it from from compassionate use cases or from lab cases. It's a really important area that we need to understand.
Thank you very much, sir. Okay, there is another question here. So from Rocky Badedeji, he said, thank you very much, Professor Hatfull, for a very insightful talk. I would like to ask if your research observed any variation, any varying therapeutic advantages from one phage morphotype to another. So was there a considerable therapeutic advantage of one bacteriophage, over another?
Yeah, so in our experience, all of the phages that have therapeutic utility for both M. abscessus and Mycobacterium tuberculosis, they're all cipherviruses. They're double-stranded DNA with long flexible, non-contractile tails. There are no phages there is no podoviruses that infect any mycobacterial strain that we're aware of. None of the myoviruses that are available infect any strain of M. abscessus or any strain of M. tuberculosis that we've yet discovered. And so, yes, that's a pretty restricted morphotype in terms of the therapeutically useful strains. It's a good question, though, and something that we're interested in, and the possibility that it could be expanded.
Okay, thank you very much. We seem to have quite a whole lot of questions here, and I think it's quite expected because of the caliber and the quality of the presentation itself. So, well, because Dr. Jessica Nakavuma is also waiting, we want to keep to time. We still have quite a whole lot of questions here. So what we will do is that for as many individuals who want to send questions to Professor Graham Hatfull, you can simply just send, by mail to Ibadan Bacteriophage Research Team at gmail.com. So I'm dropping that on the chat box now. So that makes it a whole lot easy. So that makes it a whole lot easier. So any question you have for Professor Graham Hatfull, just send it across to the Ibadan Bacteriophage Research Team mail, and then you would have all of your questions answered. So, okay, so that we will move over, we want to say a very big thank you to Professor Graham Hatfull for a very insightful session. Thank you very much for joining us. I would have said thank you for joining us on the Nigerian airspace, but this meeting is hosted all the way from the United States, courtesy of our partners, Phage Directory, Jan and Jessica. Okay, so thank you very much. Before we move over to this,